Good morning everybody. Today we are going to start with a new topic, which is about mitosis. You already know that mitosis is a type of cell division that results in two genetically identical daughter cells. We will cover this topic in this session, and you can find all the needed information in Activity 2 of Chapter 2, in Unit 1 of your textbook. First, we will start with some general information. All the cells of our body are derived from pre-existing cells, and new cells are continuously produced to ensure growth during our early years, as well as, to replace damaged or old cells. And these new cells are produced by the process of mitosis. So, what is the definition of mitosis? Mitosis is a type of cell division in which a mother cell, also known as the parent cell, is divided to produce two genetically identical daughter cells. But what do we mean by genetically identical? Do you know? We mean that the resulting daughter cells will have the same genetic material as that of the mother cell, that is the same number of chromosomes, the same type of genes and of course the same alleles of these genes. See the next example. As you can see in this photo, the chromosomes shown in the parent cell are 4 in number, shown as two homologous pairs. The first homologous pair carries two different genes. I will name them, gene 1 and gene 2. The allele capital A and the allele small a are two different alleles for the first gene, and the alleles small b and capital B are two different alleles for the second gene. Concerning the second homologous pair, these chromosomes carry a third gene, such that the first chromosome of this homologous pair carries allele capital D for this third gene, while the second chromosome carries allele small d. Now, when this parent cell undergoes mitosis, it will distribute its genetic material among two daughter nuclei, and then, the cell divides into two daughter cells, which are genetically identical to each other as well as to the original parent cell. Of course, if you think deeply about mitosis, you will find a conflict. If a cell contains 46 chromosomes, and this cell underwent mitosis by distributing its genetic material equally among two daughter cells, then, how can you explain the following? 46 chromosomes in a human cell is divided among two daughter cells and this resulted in 46 chromosomes in one daughter cell and 46 chromosomes in the other daughter cell. So, am I saying that 46 divided by 2 is equal to 46 plus 46? How can that be? To clarify this dilemma, let us look at the variation of the quantity of genetic material during a cell cycle, that is the period between two successive mitotic divisions, during which a series of biological processes take place to allow production of two new cells, because, it is highly important to differentiate between chromosome number and the quantity of genetic material. This graph represents the variation of the quantity of the genetic material as function of time during one cell cycle. Now look at the period of time just before mitosis, which is known as interphase, and this period of time includes three phases, which are, G1 or gap 1 phase, S or synthesis phase, and G2 or gap 2 phase. You can notice that the quantity of genetic material duplicated from 1Q to become 2Q. In other words, before mitosis, the genetic material doubled its quantity, so that, after division the initial quantity is conserved. To go into more details. During the first part of interphase, known as G1, the parent cell would have a certain number of chromosomes having a certain mass represented by 1Q. This cell will then duplicate its genetic material during the second part of interphase, which is S phase, to become 2Q, during G2 phase. After that, during mitosis, this quantity of genetic material is divided among two cells resulting in two daughter cells having 1Q each, which is identical to that of the initial mother cell. So, how is the genetic material duplicated? If you take a good look at the figure, you can see that, at first, each chromosome is formed of one chromatid. Let us assume that the mass of the chromosomes, when they are in such state, is 1Q. When the cell prepares itself to divide, its genetic material will be duplicated, such that a chromosome of one chromatid becomes a chromosome of two chromatids. These chromatids are sister chromatids joined at the level of the centromere. Of course, this transformation takes place during a specific period of the cell cycle, which is the S phase, and as a result, He chromosomal mass doubles to become 2q. So, at first there were two homologous chromosomes of one chromatid each, having a chromosomal mass of 1q, and after duplication, there are still two homologous chromosomes but of two chromatids each, so, their mass is double, of 2q. Please note that, the chromosomes are still non-condensed during this phase. To recapitulate, I will show you the graph one more time. But, this time, I have added some additional figures. So, you can see that during G1 phase of interphase, the chromosomes are non-condensed and are formed of one chromatid each. Then, during S phase, the genetic material duplicates, such that, at the end of this phase, the chromosomes will be formed of two chromatids, and, they will remain in this state during G2 phase of interphase. Note that G1, S and G2 phases are the three phases of interphase, that is the period of time during which the cell prepares itself for mitosis. Then, mitosis takes place, and this cell division is represented in this graph as the rapid decrease in the chromosomal mass. During mitosis, as you will see in the next session, sister chromatids of each chromosome separate, and these sister chromatids will be distributed among two daughter cells, so that, the resulting daughter cells will have the same chromosomal material as that of the initial mother cell, which was at the beginning at G1 phase. Now let us go into this general scheme, showing the changes that occur during one cell cycle, and try to answer the following questions. What is the number of chromosomes in this cell? What is the aspect, or form, of these chromosomes, during G1 and G2? What is the chromosomal mass, that is the quantity of genetic material, during G1, during G2, at the beginning of mitosis and at the end of mitosis? Is it 1q, or 2q? Pause the video, and I will correct later on, after you finish. Now let us see the answer. The number of chromosomes in this given cell is 2. During G1 phase, the chromosomes are non-condensed, and are formed of one chromatid each, having a mass of 1q. During G2 phase, the chromosomes are still non-condensed, but are now formed of two chromatids each. Their mass was doubled to 2q. At the beginning of mitosis, the chromosomes, which are formed of two chromatids, have a chromosomal mass of 2q. While at the end of mitosis, after distributing the sister chromatids among two nuclei, and the formation of two daughter cells, the chromosomal mass will be reduced to half, which is 1q in each daughter cell, the same as that of the initial mother cell. To go into more details, we will have a look at the different phases of the cell cycle, and then we will study mitosis to look at the changes that occurs during this type of cell division. The cycle is represented in this figure in the form of a circle graph. And we will talk briefly about the events that take place during the different phases. During G1 phase, the cell grows in size, and carries out normal cellular metabolism. Also, the cellular organelles, which are internal structures, duplicate in number. During S phase, the genetic material duplicate, and each chromosome would become of two chromatids. During G2 phase, the cell continues growing, and prepares itself for mitosis. Now let us take a look at mitosis. This cell division includes four phases which are prophase, metaphase, anaphase and telophase, which is followed by cytokinesis. We will start next slide by defining mitosis. Mitosis is a specific type of cell division, consisting of nuclear division, that is division of the genetic material, followed by cytokinesis, which is division of the cytoplasm. See this video to see a general view of mitosis. Now before going into the details of mitosis, let us look at the main parts and organelles of an animal cell in this next figure. This is an example of an animal cell. We are mainly interested in the cellular organelles that are labeled red. Plasma membrane, which is the cell membrane that acts as a boundary to protect the cell from the external environment. Cytoplasm, which is the fluid containing the different organelles. Nucleus, which is the organelle containing the chromosomes. The centrosome, this organelle contains two centrioles, which are responsible for the formation of spindle fibers needed during mitosis. And of course, there are other organelles that we will see in later sessions in more details, such as, the endoplasmic reticulum, the Golgi apparatus, etc. Now, to go into the details of mitosis, we will start by watching a video.
Mitosis. In this module, you will learn about mitosis. Mitosis is a process of cell division that makes possible regeneration of body parts. To elaborate, mitosis is the separation of nuclear chromosomes into two identical daughter nuclei. It is divided into four key phases: prophase, metaphase, anaphase, and telophase. An additional phase called cytokinesis occurs after the complete nuclear division. Before a dividing cell enters mitosis, it undergoes a period of growth and rest. This period is called interphase. In interphase, the genetic material of a cell increases. Prophase. During prophase, the chromatin condenses and becomes short and thick to form chromosomes. Each chromosome has two sister chromatids joined together at the centromere. The nucleolus disappears and the nuclear membrane starts to break down. The centrosomes, each with a pair of centrioles, move apart to the opposite poles, forming a spindle between them. Metaphase. During metaphase, microtubules of the spindle interact with chromosomes. This causes the chromosomes to move and align along the middle of the cell or the equatorial plate. Sister chromatids are held at the centromere, confirming the presence of protein structures called kinetochore at the surface. The kinetochore holds the chromosomes and attaches them to the spindle. Anaphase. During anaphase, the sister chromatids separate at the kinetochores, and the daughter chromosomes, along with the divided centromere, move towards the opposite poles as the microtubule shortens. The poles of the spindle also move apart and help to separate the chromosomes. As the chromosomes separate completely into two sets, one at each pole, anaphase is considered complete. Telophase. During telophase, the daughter chromosomes arrive at the pole and the chromatin starts to decoil. Also, the nuclei reform along with nucleoli. This triggers the reformation of the nuclear membrane. Cytokinesis. Cytokinesis is the partition of the cytoplasm. In animals, a cleavage furrow appears in the plasma membrane, which deepens and divides the cytoplasm into two daughter cells, each containing a nucleus. In plants, new cell wall formation is triggered at the center of the cell by the formation of a cell plate. The cell plate grows outwards till it meets the existing wall, resulting in two daughter cells. In this module, you have learned mitosis is the process of separation of nuclear chromosomes into two identical daughter nuclei.
So, to summarize, mitosis consists of four basic phases. prophase, metaphase, anaphase, and telophase, which is followed by cytokinesis. So, in prophase, the following events take place. The chromosomes condense. The nuclear envelope disappears, so that chromosomes can move freely within the cell. The centrosomes move to opposite poles. The spindle fibers will grow out of the centrosomes, and will start moving towards the chromosomes. During metaphase, the chromosomes are aligned at the equator of the cell, forming the equatorial plate. It represents the period of time during which all of the chromosomes are fully condensed, attached to the mitotic spindle from both sides, and aligned at the center of the cell, as you can see in this figure. During anaphase, the sister chromatids of each chromosome separate. This happens because the spindle fibers contract, and the sister chromatids are pulled to opposite poles of the cell. When the sister chromatids are separated, they are pulled towards the centrosomes. Note that, each chromosome which is of 2 chromatids will become 2 chromosomes of 1 chromatid each. In the last phase, which is telophase, the chromosomes are now at the opposite poles of the cells. The spindle fibers disintegrate. The nuclear envelope forms around the 2 sets of chromosomes. At the end of this phase, the cytoplasm divides by a process known as cytokinesis, resulting into 2 genetically identical daughter cells. Now, pause the video, and read this summary of mitosis. To make sure that you acquired all the needed information, take a look at how the cell looks like during the different phases of mitosis. Try to remember what happens during each phase. To make sure that everything is clear, please watch this video, summarizing the cell cycle of a cell undergoing mitosis.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into 3 discrete phases called G1, S, and G2. During the G1 or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing 2 copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has 4 stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the 2 groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of 2 genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of 4 phases: prophase, metaphase, anaphase, and telophase, and in these phases duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of 2 genetically identical daughter cells.
Hope all the information is clear enough, and you have acquired the basics. Now, let us see how cytokinesis takes place in animal cells, of course including human cell. In animal cells, the cytoplasm is divided by a cleavage furrow that forms to pinch the cell in two, as you can see in this figure. But, what about plant cells? Is it the same? Actually no. It is not the same. We have seen cytokinesis in animal cells, and it is even much clearer in this figure. You can see the plasma membrane of the parent cell pinches inward along the cell's equator, until 2 daughter cells form. But in plant cells, it is different. Look at the adjacent figure, and you can see the formation of a cell plate along the equator of the parent cell. Then, a new plasma membrane and cell wall form along each side of the cell plate, thus dividing the mother cell into 2 daughter cells. To clarify this concept, see the next video.
The main difference of the mitotic division of animal and plant cells is the lack of centrioles in higher plants, and therefore a lack of asters on the poles of the cell during mitosis. Nevertheless, a mitotic spindle still forms in plant cells, but it looks different from that in animal cells. In plants, cytokinesis takes place in the opposite direction to that in animals, from the center of the cell outwards. The dictyosome produces vesicles which fuse, forming a cell plate at the cell equator. When the plate is formed, cellulose is deposited on it and forms the cell wall. In animal cells, cytokinesis begins at the periphery of the middle region of the cell, called the equator, and proceeds inwards. This is because in animals, it is executed by a cleavage furrow surrounding the cell equator and constricting the cytoplasm.
Hope everything was clear enough. Thank you for being interested. See you next time. I hope.